Endoplasmic reticulum. Let's get started with the brief introduction of endoplasmic reticulum. The term endoplasmic reticulum was coined by Keith Porter and Francis Cullman in the year 1952. Endoplasmic reticulum, abbreviated as ER, is an expansive membranous system that pervades the cytosol from nucleus to plasma membrane and is scaffolded by microtubules. ER is the largest organelle that forms the skeletal framework in the cytoplasm of most eukaryotic cells. Endoplasmic reticulum is found in all eukaryotic cells except erythrocytes, early embryonic cells, and some egg cells. On the other hand, endoplasmic reticulum is poorly developed in spermatocytes. But in reticulocytes, it is either absent or poorly developed. The image here depicts the morphology of endoplasmic reticulum. This is the nucleus with the nuclear pore. The outer part of the nuclear envelope is continuous with the rough endoplasmic reticulum, which is embedded with tiny organelles, called ribosomes. ER is a network of membrane-enclosed sacs, called cisternae. This highly convoluted space is called the ER lumen or the ER cisternal space. This part of ER without ribosomes is called the smooth endoplasmic reticulum. ER membrane is composed of phospholipid bilayer and accounts for about half of all cell membranes. As discussed earlier, endoplasmic reticulum is composed of three components. Cisternae are a series of interconnected, flattened, membrane-enclosed sacs, tubules, and vesicles. The hollow portion enclosed by the endoplasmic reticulum is called lumen or cisternal space, representing about 10% of the total cell volume. ER also forms contact sites with mitochondria, Golgi complex, and peroxisomes. The marker enzyme of endoplasmic reticulum is glucose 6-phosphatase. To gain a better understanding of the largest organelle in a cell, let's just talk about the two distinct types of ER that execute diverse functions within the cell. Rough endoplasmic reticulum, otherwise called granular endoplasmic reticulum. Smooth endoplasmic reticulum, otherwise known as a granular endoplasmic reticulum. The main function of ER is, it is involved in membrane biogenesis. Rough endoplasmic reticulum is associated with protein synthesis, and smooth endoplasmic reticulum is associated with lipid synthesis. ER is the only organelle that cannot be obtained intact during fractionation, and it breaks down into vesicles, called rough microsomes and smooth microsomes. These pictures clearly represent the endoplasmic reticulum, existing in two forms. The rough endoplasmic reticulum, in which the membranes are studded with millions of ribosomes, giving it a rough appearance. On the contrary, the smooth endoplasmic reticulum is without any ribosomes on its membranes. Now, let's break down each of the two forms of ER, and also learn about its structure and functions. Rough endoplasmic reticulum. It is otherwise called granular endoplasmic reticulum. Rough ER is so called because ribosomes are attached to the outer cytosolic side of the endoplasmic reticulum, giving it a studded appearance. It is found throughout the cell, but its density is higher near the nucleus and the Golgi apparatus. Rough endoplasmic reticulum is specifically called Nissel's body in neurons. And coming to RER functions, RER is most abundant in cells concerned with protein synthesis and secretion. Example, pancreatic acinar cells. RER is engaged in protein modifications. Typically, sugars are added to proteins by a process called glycosylation before the vesicles transport proteins from rough ER to the Golgi apparatus. Smooth endoplasmic reticulum. It is otherwise called a granular endoplasmic reticulum. Smooth ER is continuous with the rough ER, but is not studded with ribosomes on its cytoplasmic surface. Hence, the term smooth ER. It is more tubular than rough ER, and is evenly distributed throughout the cytoplasm. Smooth endoplasmic reticulum is specifically called sarcoplasmic reticulum in muscles. SER is abundant in cells concerned with lipid biosynthesis and storage, as seen in hepatic cells, adipocytes. SER perform various important functions. Let's briefly list them all here. The primary job of smooth ER is membrane biogenesis. SER is involved in lipid biosynthesis of glycerophospholipids, ceramide, sterols such as cholesterol. Ceramide undergoes modifications by enzymes in Golgi apparatus to form complex sphingophospholipid and glycolipids. SER also synthesizes lipids of plasma membrane, endoplasmic reticulum, Golgi complex, lysosome, nucleus, peroxisome, and mitochondria. SER synthesizes fatty acids and is involved in desaturation of fatty acids. The second major function of SER is liver detoxification. Detoxification is the process of converting the harmful and insoluble substances into harmless, soluble, and excretable forms. Sarcoplasmic reticulum acts as intracellular storehouse of calcium, which enables muscle contraction in muscle cells. Other functions include synthesis of steroid hormones from cholesterol, bile components from cholesterol, prostaglandins, and vitamin C, that is ascorbic acid. SER is involved in several metabolic pathways such as gluconeogenesis, glycogenesis, glycogenolysis in liver of fasted animals, and uronic acid metabolism. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.